Okay. Am I on? Okay. Am I on? Okay, good. All right.
John Harbaugh! Silence! Silence, I say!
Well, still 1 minute according to that clock. That clock is wrong? Okay, let's get started. Huh? Okay. All right. Okay, let's get ready. Still got 1 minute. Okay, let's see.
We're not streaming today, so we're just recording.
Oh, just recording? Yeah, that's good.
We hope so.
Yeah. Let me turn on my recorder. Okay. There's my recorder. Okay. Let's get started. Welcome, everybody. This is the Silicon Valley Fourth Interest Group, April 28th. Okay, and I'm Dr. Chen Hansen Ting, and my subject today is microRNA and long non-coding RNA. So I think for all you computer hacks, this is kind of biology 101. So let's get to it. So this morning, what I'll be talking about is microRNA, which is short for miRNA, and long non-coding RNA, which is usually shorted as LNC-RNA. Okay. And the theme of this talk is that microRNAs are really tokens of the cell computer. So I try to prove or just get some indications that this thinking is in the right direction. So I will show you the results of my work in matching lots of microRNAs into the long non-coding RNA. And interesting enough that I would show you that some of these clusters of microRNA in long non-coding RNA looks like fourth code. Okay, so I have the kind of a belief that I have found the real code in long non-coding RNA. And I would make a comparison of fourth computer and cell computer. So I think these two, the cell computer operates very similar to a fourth computer, but of course in a much more complicated way. So this is the topics I'd like to talk this morning. Let me just give you, computer hackers a brief introduction on the microRNA. MicroRNAs are small RNA molecules which generally has 22 nucleotides. And it's been found in plants, animals, and in viruses. The functions in RNA is, so far, we have seen a few RNA whose function has been identified. And their function are basically mostly in silencing and post-transcriptional regulation of gene expression. That means that the gene has expressed into messenger RNAs. And some of the microRNA can silence some of the messenger RNA, thus deactivates the process in producing proteins. Okay, so there has been many people studying microRNAs, and many Nobel Prizes being awarded to people working on the microRNAs. So there are organizations which collect lots of microRNAs and release them on the web. So miRBase is a searchable database of all published microRNA sequences and annotations of many species. It's maintained in this website. So lots of information about microRNA already available. So in this database, 48,000 microRNAs being collected. And so I went through this whole thing and found that there are 27,000 unique microRNAs. And in human RNA has, in this database, it has listed about 5,000 human microRNAs. So we have a big database, lots of sequences, but very little understanding on the whole collection. So this is the start of this database that I downloaded. So basically, I'm showing you these microRNAs which were discovered in human. So you can see that they have giving them identifications and giving them these are the old identifications, and right now in this database they just try to sequence everything so that they have a uniform uniform identification. And here are all these various sequences. Some are similar, but not necessarily identical. So in this database, I have tried to clean out all the duplicates. So and it's interesting that many different species share the same microRNA. Lots of microRNA occurred in many different species. So my assumption is that I will just use all the database, all the microRNAs listed in this database as my studying objects, and regardless their species, regardless their origin, because I assume that all the living organisms share the same set of microRNA. This may be true, may not be true, but this is a good working assumption, because I've seen lots of microRNA species, lots of microRNAs are shared by many different species. Okay, so this is all the data I've collected, and I will use them for my analysis. Okay, now the next thing is the long non-coding RNA. So this long non-coding RNA are RNA transcripts which are longer than 200 nucleotides that are not translated into protein. They are found in between coding genes. Okay, so there are lots of long non-coding RNAs discovered over the years for all different species, and these long RNAs that's discovered in different cells, and they have sequenced them, and found that they do not correspond to any known proteins. So they are named non-coding RNA because they do not code for proteins. Because most of the protein people have studied are used to encode proteins. These are the messenger RNAs. And RNAs which are not messenger RNAs are all collected, known as long non-coding RNAs. But my point here is they are coding RNAs. They have codes in it, okay? And I want to demonstrate to you that they are coding it. So this non-code is a database which try to collect all the long non-coding RNAs in one place. And they have collect RNAs from many different species. I think that in the current state, they have collected long non-coding RNAs for 17 different species, including human. And this database is maintained at this website, and you can go look at them. And if you are interested, you can download some of the code. So I look at it, and I'll just download the human part. So I think human in this collection, the human long non-coding RNA is the biggest collection. Okay, so I thought that if I want to study anything, the human long non-coding RNA could be a good place to start. So the human long non-coding RNA in that collection has 172,000 transcripts, and they have identified 96,000 genes in it. I don't know exactly what they mean, okay, but they are not coding for proteins. And the total size of the file is 284 megabytes. Okay, so that's a huge file, and I was kind of attracted to it. Well, let me see if I can actually read it. Okay, I can actually read it, and I can also import it into my program and can see what's in it, and doing some edit on it, and doing some interesting work. So this file is not a deterrent. I can handle it. But if you ask Windows Word to read it, it just spins, and would not actually give you anything. Okay, so it just stuck there. The file is too big for Word. So here, this is the beginning part of the human long linear RNA. You can see that the database is organized with one line of header, and then followed by many lines of code. And this goes on for each transcript in the database. It has this kind of organization. So what I did was, okay, here is some statistics. This is from the human.fa file. This is the DNA data file in text form, in annotated. So it has the headers. So it's 284 megabytes. So the first thing I do is to split this file into a data file and an index file. So I want to collect all the data in one file, and then all the pointers and the index, the headers in another file, so I can know where each RNA transcript is at, and I have the data, and this data, I want to strip out all the character returns and line feed, and all the characters which are not code. So in this file, I have pure code, okay, from the start to end, because that's the data file I can analyze. I don't have to worry about the things, the text messages interdispersed with the data. So I split it into data file and index file, so I can access them more conveniently. So the microRNA collection, I have the mature.f, which has 48,000 entries. So I sorted, I think 48, 3 megabytes of data. Well, Microsoft Word will read it very reluctantly, okay? And even Excel, you can force Excel to read that file. And once I read it into the Excel, I can separate the data into columns, and then I'll sort the entire file according to their gene sequences. So once I have sorted the file, you can identify those which are redundant, because if you sort the entire file according to their basis, they'll line up with all the bases, and if there's any replicates, they'll just appear one next to the other. So I go through it and delete all the duplicates, and I end up with a file with 27,790 microRNAs, and the file is to 843 kilobytes. So what I want to do, I have a big collection of long non-coding RNAs, and I have a big collection of microRNAs. So what I want to do is to search through all the data in the long non-coding RNA to see if there's any microRNAs in it. And I'll try to identify all the microRNA occurrences in this big data file. And then I just print out those matches, okay? And I would think that if I can find anything useful, then I will see how many microRNAs can be identified and how these microRNAs are arranged in the long non-coding RNA. So the basic idea, basic premise is that each microRNA is a token, just like in the fourth word. A fourth word generally will have a name and a token, and this token indicates how this word will be executed. So the microRNA, I'm thinking that it is just like tokens in a fourth computer. And the microRNA has a very nice property in that it has 22 nucleotides, okay? So each nucleotide has 4 different options, A, G, A, C, G, T, okay? So each nucleotide can encode up to 4 bases. So each nucleotide is worth 2 bits. Okay, you have 4 different choices. So 22 nucleotide, that's equivalent to 44 bits. And 44 bits can encode 16 trillion tokens. So the microRNA as tokens, it has a very wide coding range. And for tokens, you need tokens to be unique. Okay, you don't want tokens to be duplicates. Then it just confuses the computer. So the computer would not allow duplicate tokens. If tokens are the same, they must have the same function. Okay? So this microRNA is big enough to encode all the functions that the cell computer would have to perform, right? So it's big enough. It's good enough for tokens. Okay. And another very interesting thing, it has short lives. Okay. And this is important because if you have a microRNA which lives forever, so it will do its function forever. And that may be good, and most likely may not be good. So a short lifetime of microRNA is very essential for the function of a cell computer. So cell computer, if it wants to do something, it will issue microRNA. It goes out and do its things, and after it performs its function, it died. Okay, it's no longer there, and so the cell can do other things. So it's big enough to do code. It has short lifetime to be functional, but don't live forever. And it's small enough to send signals to other cells. So these microRNAs are small enough they can penetrate the membrane, the nucleus, and go out into the cell. And it can also penetrate the cell wall and get into the body fluid and goes to other places and do other things if the function will have to be done with a collection of cells. So I think this microRNA has the right properties for tokens to support a cell computer. So this is my work. So I want to match all microRNAs to all long non-coding RNAs. So first, I have the human long non-coding RNA, and I compare I search the data in the human long non-coding RNAs for all the occurrences of microRNAs, which I collect 27,000 of them collect in my microRNA database. So in searching all the 171,000 long non-coding RNAs in the discovered in human being, I found 22,651 matches. Okay, so there are between these two data files, I found 22,000 matches. And this file, resulting file is 391 kilobytes. So there are two most frequently occurred microRNAs. M17325, which occurred 7,000 times. And then M26622, which occurred 5,000 times. So I delete them because I don't know what they are. but they occurred so frequently, must have very important function. But what they are doing, I don't know. Okay, but this is what I see. If I do the matches, I found the matches. This guy matches the most time, and this one is the second one. And then there are many of the microRNAs which occurred more frequently than the others. So there is a distribution, but these two are the dominating entries. So what I did was that I sorted this this file, this long non-coding RNA file, and then I pick up all the matches which are in a sequence. So if I combine this file with the index files in the RNA, the headers. So if I put headers back into the searched match list, they form the header just insert into the sequence where the long non-coding RNA begins. So after this one inserted, you can see the whole thing is like a fourth program. Okay, you have headers, which are the indexes, and then if one of the long-coding non-RNA has some entries, so the entries would be just following the headers. So you got pretty much like a dictionary in fourth. So I want to find, there are individual matches. They are interesting, but that's not a pattern I wanted to find. What I want to find is a collection or clusters of microRNA which occurs right next to each other. So there are many in this collection. I found 22,000 matches, but several of them are long stretch of microRNA entries, and these are one next to each other. So you see, microRNA, they are generally 22 nucleides long. But here you can see they are sequenced 38, 79, and 37. These are the reasonable spaces between microRNAs.
Some of those columns, what are they?
Okay, the headers are the indexes. You see,
The first column is the index?
Yeah. The top one is the index. Okay, the M is the IDs for the microRNAs. P are the IDs for the long non-coding RNAs.
What about the column on the
This is the address.
That's it. Okay, this is like the offset into the...
This is the address of the data file.
Okay.
Okay, so these many microRNAs occurred consecutively in the data file of the long non-coding RNA. And this stretch is following the header of the T3467. So these are microRNAs probably appeared in this long non-coding RNA. But it is not directly from the beginning. You can see in the beginning there's a 808 basis stretch, which doesn't have any matches. Okay, and the matches start from there, and it continued to have a whole bunch of them. Okay, and I think that's what I'm looking for. Okay, I think if this these microRNAs are tokens, it will be very very. If it is a fourth program, I'm expecting tokens to be consecutive. Okay, and this is what I found. Not many of them. I have 22,000 matches, but only a few stretches, which has consecutive microRNAs, and so. Okay, so here this is the actual data associated with that long non-coding RNA or a gene, and then you can see that about 800 code doesn't have any microRNAs in it, and then suddenly you have lots of microRNA stitched together, okay, very nicely into a list, almost a list, okay. So that's what I think. If this can be a program, okay, and the program consists of consecutive microRNAs, and which this list is most likely invoked somehow. And then once this list, once this long non-coding RNA is invoked, most likely it will release this set of microRNAs. I don't know if this microRNAs will be released in sequence or with certain time delay. I don't know. Okay, but it could be that they are released all at once, and each of them go out to do their own things, and they might invoke other long non-coding RNAs to do much more complicated things. Just like a fourth program, we have colon definitions, and we collect all the colon definition into a high-level colon definition, and then build up to a very high level. And once the high-level colon definition is called, it will just execute the list of functions it compiled previously. And each of these entries may call another list, and the list call down until you find a token which was encoded in machine code, and you execute the machine code. Okay, and in here you go the list down and down and down. Finally, you meet some microRNA which encodes for protein, and then you produce the protein and do the function that it is called for. Okay. So far? Yeah.
Well, it's interesting. You're saying that the microRNA are like the nucleus, the token.
Yeah.
So that these other things would be like higher level words that are in terms of other tokens.
Yes, exactly.
Yes.
Mm-hmm. That's fourth, is it?
Oh, well, actually, aside from them being sort of consecutive, like what percentage of like how common are you finding matches?
Okay, so far, you know, you have.
Probable in the first place.
Yeah, this right, because although we have 50,000 microRNAs discovered over the years, and for the human being, we have 171,000 long non-coding RNAs identified and recorded, collected in the database. We don't know if these are the complete set, okay? we're probably far away from the complete set. So we have a very small collection of microRNAs known, identified. And we have a very small collection of long non-coding RNAs. So the matches, you don't expect they would match 100%. Okay?
Well, I'm saying just even given the
the
search, like the size of 22.
Yeah.
That seems like that's a pretty low probability.
Yeah, that's right. That's right. That's right. The matches, statistically or randomly, the probability of match is basically 0. So, by the fact that I've discovered 1 or 2, or I discovered in here, I think variable length, I think I've identified about 10 sequences that I think is worth paying attention to. I'm sure I don't have the complete set of microRNA, so there's a lot of gaps. But I think over time, probably we can fill in the gaps and find the whole things, maybe completely from the beginning to the end, lists of microRNAs. Okay? So let's, well, this is for my friend. I invite him to hear fourth computer. So what is fourth computer? Fourth computer is a Turing machine, which has a set of commands. So it has a set of commands. And these commands we call tokens. Each token has a name and a function. Each token has a name and a function. So the name is important because with the name we can search. A human can search this command, and if we find this command in a computer, then we can execute it. So name is very important, but of course the function is more important because that's what the actually how actual works are done. So there are two and forth. We have several different types of commands, but the most important two commands. One is a primitive token. The primitive token, it executes machine code. Okay, so you actually do things in a real computer. That's the primitive token. And another set of tokens we call high-level tokens. These tokens executes a list of tokens. Okay, and the tokens in this list can execute another list of tokens. And this another token of list have many tokens, and each of them may call another list of tokens. So you see this structure is layered, okay? And the tokens can call list of token. Tokens in the list can call another list of token, and until at the lowest level, tokens are built-in. Tokens are executing machine code, and which performs real work. So this is what a fourth computer is. And the high-level fourth token, it is a token list. Okay, and basically when we compile it, we use this syntax. So you have colon, and you give a name. This name, you can imagine any kind of ASCII sequences. And then you give the names of other tokens, okay? And then you terminate with a semicolon. So this builds a list in the computer. And when this name, this command, when the name of this command is invoked, it would go out and find this token list and actually executes this token list. And this token, each token may call another token list, and another token list may call another token, and all this thing. But in the end, it reaches to primitive instructions, and they execute machine code. So here is an example. So dupe, multiply. These are two basic forth functions which duplicate number and then multiply them together. So this makes a square. Okay, so if I want to do cube, I want to take a number to the third exponent. So we duplicate, do a square, and I have another copy of the original number, and I multiply it. So I multiply the same number 3 times and get its cube. So if I wanted to take it to the 4th power, and I just do square and do the square again, this square was square of what the result we got before, so I get power of 4. Okay, so this is how we write forth programs. You have a list, give it name, and you have a new word. And so we have a square, and the square can be used in another word to build another big word, and this word can be used as elsewhere to build even bigger words. Kind of clear or still muddy? Yeah.
Okay, as you know, words
Yes.
have a field, there's a link field, a field, there's a data field, but there's also a code field.
Yes.
That would be interesting to be able to find the equivalent of that in DNA.
Yes.
Thing that says,
Yes. Yeah. Right. But we don't know. Okay. I can.
in fact, the DNA may be headerless forth.
Yeah, it's headerless.
Right.
We don't have any header. No names.
Just
Yeah. I think it's just we just have the we have the DNA, the big long stretch of code, and we have microRNAs. MicroRNA has 22 nucleotides, and we know this microRNA can go out and find a match in the DNA. So experimentally, we have proven that if you have a pattern in a small stretch, and this stretch will just match exactly the code in the DNA. Yeah, found that, and then something interesting will happen. Sometimes, we know it can duplicate. It can do the polymerase.
yes.
If you give a new one point in the DNA, and then the polymerase will come starting from here. It just make a copy of it. Yeah. So that's it's the it is both code field and address. The DNA does not know the addresses. Okay, it's very long. Each different chromosome has variable length, and they are positioning it, but they are not marked with address, but they are marked with specific patterns of nucleotides. Mm-hmm. Content addressing. Yeah, the content addressing tokens. Okay, the tokens has its code, and this code most likely are used to match patterns in the DNA chromosome and find that specific location, and then it would do the copying or do the called the transcript. A transcript, a stretch of information into the messenger RNA, and the messenger RNA will be used by the or whatever the mechanism to produce a new protein. Okay, so here is the a very brief introduction to fourth, and here is what I envision. What is a cell computer? So, cell computer is Turing machine, and it is using microRNA as tokens. Okay, and we have a set of primitive tokens. What they are, we don't know yet. Okay, but this set of primitive token, each token will activate one messenger RNA, which produces one protein. So in a cell, each messenger RNA must have a mechanism that can be used to activate it. How? I think it is a microRNA. Probably this microRNA in some form exists in front of the messenger RNA code. And when the microRNA found it, then the subsequent messenger RNA gets transcribed. Okay? But this is only a very small set of microRNA, because currently, I think current thinking in the industry is that human being needs to make about 20,000 proteins for its entire function. So 20,000, if human being needs 20,000 proteins, we've got to have 20,000 microRNAs. Each of them commands the producing of one protein molecule. And but most of the coding RNA, that's the coding RNA, which produces protein. But this is only a very small part of the entire DNA library. In human being, they have say, well, it's only about 1.5% of the entire genome is dedicated to protein manufacturing. We got the 98.5% of so-called non-coding DNA. But I believe this really, this set of microRNA which produces protein is a primitive microRNA, which is only a very small set of the microRNA, and the majority of the genome contains tokens which would activate other tokens, and other tokens would activate a long non-coding RNA, and this long non-coding RNA would activate a sequence of microRNAs, which causes a sequence of events, and which eventually perform the function required by that high-level token. So most tokens activate linear long non-coding RNA to produce other microRNAs to perform more complicated tasks. Yeah.
MicroRNA that creates proteins.
The messenger RNA.
The messenger RNA.
Yeah. Mm-hmm.
Would there be an RNA? I'm thinking, suppose you have you're building like a building, you have a framework, right, that you use to build the building. Then when you're done, you want to take that framework down. So would there be messenger RNA to destroy protein or break protein down?
Yeah, there are protein to break protein down.
Oh, okay.
Yeah, there are protein which are catalyze. There are enzymes to catalyze different proteins. Yeah. They clip and do the edit things. Yeah, there are lots of protein doing all these very complicated things, but so far I think we have identified probably 10,000 of them.
And there's probably a whole lot more.
Not a whole lot more. Okay, the first thing that they have, 100,000 of them, but now the consensus is that we have about 20,000. That's it, and the rest of it, the rest of the DNA, what are they doing? They are high-level codes. Okay, so the coding DNA, what we understand as coding DNA, are actually low-level primitive code. Low-level primitive code, and all the more complicated functions are in the non-coding DNA, and they are most likely expressed into lists of microRNAs, and this list of microRNA calls other lists of microRNAs, and then perform very complicated functions. Okay, I think this is my thinking on how this cell computer works. Okay, so most long non-coding RNA contains microRNAs. Some non-linear RNA have lists of consecutive microRNAs. Some primitive microRNA activates messenger RNA to make proteins. Some microRNA activate long-linear, long non-coding RNA, and which invokes a whole set of other microRNAs, and then do all the things. But definitely there are microRNA which does functions we don't know yet. Okay, because we really don't know what all these non-coding DNAs are doing in the cell. Okay, so in the human long non-coding RNA, these are many of the sequences I have uncovered, and I can write them in Morse code. Okay, so here codon definition. This is a long non-coding RNA, and it has a some microRNA, and that's a stretch. I don't know. Okay, I don't have match. But then following that, there's two more, a stretch, and then some more. Okay, so there are quite a few occurrences where you can see lists or clusters of microRNAs. Yes.
How about this? In the non-coding. Okay, you came with this list of guys here.
Yes.
Do you think you might be able to find references to say like T34667 in the coding?
I would like to see it. I would like to see it. I would like to see it. Yes.
Yeah, because it's like.
Yeah.
Well, that's part of this word. Because as you know, you get to the higher complement.
Yeah.
You have basically just a row of CFAs.
Yeah.
Say jump to here.
Yeah.
Okay.
Yeah. Okay. Let me just comment on it. You can see. You hit upon a very important thing in this coding scheme, because you see we have lots of microRNAs here. Okay, and how do we call this one? Okay, there must be something in front of it, which is microRNA. So this microRNA here, it got the match, and it produce this thing. But
Right. It's like you're looking for a colicys.
yeah. So here are the pointer field. Okay.
Yeah.
So you have to find this pointer, and then you get the stretch. But how about this one? So if I have a microRNA here, can I jump directly into here and make the match? And what happened? Okay. So this is very important, because in the long non-coding RNA, these codes are not visible. They are smudged. Okay. They are not available for searching and matching, because they all form what they call the helping. They have helping structures. So this RNA wraps up into a helping with some code following it, which matches. So these are all in the helpings. And if they are in the helpings, they are not available for matching. Okay? So you see here in the code, they are hidden from searching because they form helpings. And so only those which are not in the helping is open for matching. Okay, let me get. This is very interesting, but I think let's move on. So microRNA in genomes, if microRNA clusters appears in long linear RNA, they should also appear in genomic DNA as well. Okay, so because long non-coding RNA, they are transcribed from DNA. So the information in the long non-coding RNA must exist in the DNA as well, because that's where they are. That's where they are stored. Okay, so genome library are more readily available for many species than non-coding RNA. So we got the long non-coding RNA library. Even we have a very big, huge library for the human thing, but we know they are not complete. Okay, so the complete information, where are the complete information? They are in the genomic DNA. So instead of searching microRNAs in the long non-coding RNA, why don't we just go to the original source to search the genomic DNA? Yeah. So that's what I'm doing. I was very encouraged to find long stretches of microRNA in long non-coding RNA. But I think we'll find the same thing or a very similar thing in the genomic DNA. So I think I don't have to be limited by the long non-coding RNA library. So if I have the original complete DNA genome libraries, that's the best source for searching and matching. So when I've done, I have downloaded the mitochondrion, and I have talked about it in an earlier session. So mitochondrion is a very small organism in cell which produces energy for all the cells. So its genome, it has an independent genome passing from mother to the passing only from the mother to the offsprings, and it is a very small stretch of DNA. So I said, well, why don't I search that one to see if I can find any microRNAs in it, because the mitochondrion inside the cell has to be controlled somehow. Okay, but it's too small. the non-coding part of it is too small to have a significant program in it. So I was looking at and I couldn't find anything in it. And I think, if I can find some microRNA in it, that solves the problem, because then the cell can send a microRNA to a mitochondrion organism and cause something to happen in the mitochondrion. So cell controls mitochondrion by sending microRNA to it. There must be sites in mitochondrion genome to receive microRNA signal, right? And last night I found them. Okay, so I actually found using the microRNA library to match the mitochondrion DNA and found 5 matches. Very, very interesting. Okay, and this 624 is very close to the D loop, the control loop. So I think this M16915 might be the critical mitochondrion, critical control mechanism for mitochondria. But these other things may also be important. I don't know, but there are 5 microRNAs identified in the human mitochondrion. So that's very exciting. And then, of course, the model organism in the genomic study is the C. elegans. And lots of Nobel Prizes were awarded with this small worm. It's called a flatworm. Okay. Yeah, this is a flatworm. Yeah. Mm-hmm. And in 2002, Sidney Brennan got Nobel Prize on his work in this organism, because he identified a couple of microRNAs which controls the development of elegans from stage. I don't remember how this organism is developed, but to evolve from one stage to the next, you have to have this microRNA. Okay, and I think he got the Nobel Prize for that. So the microRNA controls the development and the death of elegans. So it's interesting to see how, if we can find microRNAs in elegans DNA. And there you are, found a beautiful cluster, okay? And that's occurs somewhere. It's interesting, this numbering. This numbering random, but because Brenner was the first one, he pioneered the study of microRNA. So the microRNA he discovered were generally listed first, okay? So he had number 6, number 9, number 10, number 11, and number 12, all sequence. These are the microRNAs he first identified, okay? Then his postdocs and his students keep on working, and they identify more of the microRNA. So you can see this 2000, 303, 2003, 04, 305, 306, 307. And these are, I think, discovered by his students. Okay, and they some year they published it, and so they collected and gave consequent numbers. And all these things just happened to be in one stretch of RNA in the elegans genome. So here, its location is here, and we have a stretch of it. So that's very, 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 very interesting. So I just show you how these things are arranged in this stretch of DNA. Okay, Conclusion, cell computer uses microRNAs as tokens. I propose, okay, this is my postulate, okay, and there are two different classes of microRNA. The primitive microRNA activates messenger RNA to make proteins, and high-level microRNAs activate clusters of microRNA to perform complicated tasks. If you see the code, if you see how clusters are arranged, I think you can inevitably conclude cell computers are force computers. Okay? We don't know everything yet, but we have indications that this is the case. It looks like God designed the universe using the simplest laws and applied them in the most complicated ways. Okay, that's what we see. Okay, it looks like that God designed the cells using the simplest force language and applied it in the most complicated ways. Okay. Any comments so far?
Well, I hope you're not trying to say that God's a nerd.
He is, okay. He is a very good mathematician.
Oh, yes.
Okay.
Exactly.
And the quantum equations, we have the quantum equations. We knew quantum equation worked because it worked on hydrogen atom. So we believe it worked on all other atoms. But we cannot solve the quantum equations for any other atom, any atom other than hydrogen.
It has to be done by approximation.
Yeah, that's cheating, okay?
Well, I know. You go to the rest of the stuff, or in astronomy, what we call metals.
Yeah, really, that's all. Okay, and I think this is, I think for immediate future, this probably is the most influential conclusion. This microRNA therapy. So microRNA must be therapeutic. So the future therapy will be done by microRNA profiling. So you go to a doctor. So he just, I don't know how, he just look at all your microRNAs. He doesn't have to look at all your DNA, but he probably would look at all your DNA so that he can derive all the microRNAs. But he has to profile what you have currently. And from that profile, he may find microRNA deficiency. Certain microRNA is not there. And therefore, you get disease. So if this microRNA deficient disease can be cured by microRNA administration. So just find out what microRNA you don't have, but you should have. He just picks up one needle and stick it into your arm, and you're cured. But there are other microRNA which might be overexpressed, which causes problem, like cancer. Okay, yeah, that's because microRNA just run amok and doing all its things and grow the tumor instead of your body. Okay, so for that kind of disease, he might find a specific microRNA which can suppress the overexpressed microRNA. And I think very easy to do, because you just have to give it the anti-microRNA. You just give it the microRNA which is exactly complement of the microRNA which causes trouble. And this microRNA will just adhere to the microRNA which causes trouble and defeat it and cancer is cured.
Choose complement.
Yeah.
There you go.
Right. Okay, okay. I think my time is up. Yeah, yeah.
Are you going to publish in this?
Well, I'm publishing, right? You hear?
Well, this is published, you know, like.
You're going to send your slides in to Dave?
Yeah, sure, sure.
So what I mean is, like I assume there's a mega, a publication like Genomes are us.
No. Well, there's lots of things, but I don't think I can. Anybody would accept my paper for yet. Science, nature.
Nature? That huh?
JAMA. And Scientific American. Okay. Yeah.
I'm a postdoc in the medical school, so I'm working on the same field. But yeah, it's a great hypothesis. But I'm wondering, in the force, is there some code to organize which token is executed first and then second? So to organize these tokens, and, because I think this microRNA has some sequential, maybe in a different different location.
Yeah.
This one is first.
Yeah. Right. In force, the sequence is very important. Yeah. So things has to be occur sequentially. Okay, but here I don't know.
It could be parallel to some level.
Yeah, it could be. There
must be some. What you're saying is like an RTOS, time operating system.
Yeah.
Somehow it's
there. Yeah. It could be executed sequentially with certain time in between. Of course, if the time is very short, it just instantaneous. Okay. But it could be long delays, and it could be that some feedback is required for getting into the next step. Okay, so these are all possible, but from the code itself, we don't know. Okay, we found the sequence, whether this is in the physical sequence. We don't know, or it might be all at the same time. It's still possible. It's released all at the same time. So each of them just go to the different places and do the different job.
It's also what you need to also find in there is the outer and the inner force groups.
Interpreter, compiler. Yeah. Well, the God didn't give us the compiler. He gave the object code. Okay. Okay. You only have the object code. Yeah. So what I'm trying to reverse engineer what he.
What basically God is saying is the work is left to the student.
No, I don't know. I don't know.
You'll figure it out.
Yes, you go figure. You go figure.
That's why maybe.
Well, this is a headerless forth, so
yes.
Basically the names of the words are code, right?
Yeah.
There's no name. There's no name as we noted. Yes, right. I think the addressing is by content addressing.
Content of
the microRNA.
Which is also the code. Right?
Yeah, the code. Yeah.
So it's kind of like a while ago I gave that like parse time execution document.
Okay.
Where I have the code as the name of the word. I guess.
Yeah. That's right. We can just use the code as the name.
I wanted to be sure everybody realized we're not going to go to the Hoover Pavilion because the exhibit that I was touting doesn't start until May 24th. So we're not going to be going over there.
Okay.
Today.
Good.
We're going to lunch.
Maybe in June.
Yeah, Dave. Okay, let me.